They scanned human DNA for weakness and found weapons. In the early years of the 21st century, genetic research advanced at an unprecedented rate. The Human Genome Project had mapped the entirety of human DNA, providing an intricate blueprint of our species. But what started as a mission to understand diseases soon took a darker turn. Dr. Evelyn Carter, a lead geneticist at the International Genomic Research Institute, IGRI, had been working on a project designed to identify genetic vulnerabilities that could predispose humans to certain conditions: cancer, neurodegenerative diseases, immune disorders. The project aimed to create personalized treatments, but hidden within the data was something far more alarming. Patterns emerged that suggested specific genetic sequences weren't just weaknesses, they were potential biological weapons. DNA contains the instructions for life, but certain sequences are more susceptible to manipulation than others. Carter's team found that variations in genes responsible for immune responses, cognitive function, and physical endurance could be selectively triggered. If exploited, these variations could incapacitate a person, or an entire population, without the need for traditional weapons. One discovery stood out: a sequence within the HLA, human leukocyte antigen, complex. It controlled immune system efficiency, and small alterations in this region could trigger autoimmune diseases on demand. In theory, a precisely engineered virus could activate these weaknesses in a targeted manner. Another unsettling revelation came from the FOXP2 gene, which regulates speech and cognitive ability. A well-designed genetic attack could degrade neural pathways, leading to speech loss, impaired decision-making, or even complete mental collapse. It didn't take long for governments to take notice. Initially, Carter's research was meant for medical purposes, but intelligence agencies soon stepped in. The potential to develop genetic weapons, silent, precise, and undetectable, was too tempting to ignore. Countries began secret programs aimed at tailoring bioweapons based on genetic susceptibilities of rival populations. It was no longer about mass destruction, it was about precision targeting. By analyzing genetic databases, nations could identify specific vulnerabilities in enemy populations and design biological agents that would affect only those with a particular genetic marker. This was the dawn of precision biowarfare. Dr. Carter, horrified by the turn of events, attempted to halt the research. She had entered the field to heal, not to destroy. However, the genie was out of the bottle. She went public with her findings, triggering an international debate on the ethics of genetic weapons. The scientific community was torn. While some condemned the weaponization of DNA, others argued that if one nation had the capability, others needed to develop countermeasures. Genetic counterweapons, designed to neutralize hostile bioagents, became a new arms race. Despite government efforts to contain the information, a rogue scientist leaked classified documents detailing how genetic weapons were being developed. The world was unprepared for what followed. Small-scale attacks began surfacing, targeting specific individuals in political and military circles. Untraceable illnesses affected high-ranking officials. Intelligence agencies scrambled to identify whether these incidents were natural or engineered. The world entered an era of genetic espionage, where DNA databases became national security assets, and personal genetic information was the most dangerous currency. Realizing the irreversible nature of their discovery, Dr. Carter and a team of ethical scientists formed a resistance. Their mission: to develop a universal genetic shield, a defense mechanism encoded into human DNA that could neutralize malicious modifications before they took effect. They worked in secret, using CRISPR technology to create a genetic firewall. But with every step forward, opposing forces tried to dismantle their progress. The battle was no longer just scientific, it was a fight for the very essence of humanity. As the world teetered on the edge of a new form of warfare, the question remained: could science be used to undo what had already been set in motion, or had humanity's knowledge of its own DNA become its greatest curse? With more nations entering the genetic arms race, new discoveries emerged. Scientists began uncovering additional vulnerabilities in the human genome, including sequences linked to psychological resilience, metabolic function, and even pain tolerance. Bioengineers speculated that an adversary could create a virus that selectively amplified stress responses, driving entire populations into states of chronic anxiety and paranoia. The military-industrial complex took an interest, pouring billions into research on both offensive and defensive genetic modifications. New programs aimed at enhancing soldiers emerged, creating individuals resistant to genetic attacks while increasing their endurance and cognitive function. The ethical line between defense and enhancement blurred further. While governments waged their secret battles, biotechnology corporations saw an opportunity. Several companies began collecting genetic data from unsuspecting consumers under the guise of health services. By offering ancestry tests, personalized medicine, and even gene therapy, these corporations amassed massive DNA databases. Many were secretly contracted by defense agencies, providing crucial genetic insights that could shape the future of biological warfare. Dr. Carter, now a fugitive, attempted to warn the public, but her efforts were met with resistance. Governments and corporations alike sought to discredit her, labeling her a conspiracy theorist. Yet, a growing underground movement of scientists and activists rallied behind her, seeking to expose the truth. As engineered genetic modifications became widespread, an unintended consequence arose: mutations. Some populations exposed to gene editing viruses developed unforeseen genetic resistance, rendering certain bioweapons ineffective. This led to an escalating cycle of adaptation, where both sides, attackers and defenders, raced to stay ahead. A radical faction within Carter's resistance proposed a controversial solution: rewriting human DNA on a fundamental level, erasing vulnerabilities altogether. But such an endeavor carried risks. Could they unknowingly create a new genetic elite while leaving others vulnerable? With tensions at a breaking point, an international summit was called. Scientists, world leaders, and ethicists debated the future of genetic warfare. Carter, risking her life, infiltrated the event and presented undeniable evidence of the ongoing biowarfare programs. The revelation shook the world, leading to global protests and demands for regulatory oversight. A fragile treaty was brokered, banning the use of genetic weapons. However, history had shown that treaties were often ignored in times of war. Carter and her allies knew that their fight was far from over. They continued their work in the shadows, developing countermeasures for a battle that might never truly end. As she looked to the future, Dr. Carter understood one truth: humanity had unlocked a power it could not control. The only question was whether wisdom would prevail before the next genetic war began.